Hi, good afternoon. Can you hear me? You need to unmute yourself. Uh, but if you cannot speak, it's fine. You can just write in the chat or you can just connect only. We will wait a little bit to maybe other people will join. Good afternoon, Anish. I think everything is good with you. We will wait a little bit to others to join. Okay. Okay, that means sound is working. Thanks. Uh, I think there was some other people also. Sorry, I missed that. I was messaging others to join. Um okay, we can quickly start, and if after that still a lot of people wanted to join or something, please let me know. I will share my screen. Um, so, Mice, how is everything with you? Is everything all right? Um, uh, if you guys wanted to speak, you can unmute yourself and speak. If not, it's fine also. Uh, okay, just quick question. Our podcast 1, 2, 3. Did you guys listen to that? You can write in chat or you can unmute yourself and let me know. Um, and also, because I think I could not share, I was a little bit busy with some other stuff. So I just shared the link. So if you know anybody else who will be available to join the uh, group study, please let them know. And also, I think if you go to our Discord, if possible, there's some of the people I think is still over there. You can just knock them and let them know that we are doing a study, so they can join. Okay. And in the meantime, if anybody cannot join or something, if they're messaging you, just let me know. I will let them enter. Um, um, I think without further ado, we can start. It should not we should not waste others' time. Um, and the thing is, we cannot record this group study because we gonna discuss raw data, raw stuffs. Um, it will be better to just like this kind of discussion. It's better to like. Uh, Hi, good afternoon, Dr. Mohit. I was waiting for you. I think you can hear me, right? If you cannot unmute, you can just write in the chat or just message me. So I think everything is fine. So I'm gonna quickly share one screen. We're gonna see this is a NGS of like it will be in general discussions. Then, if anybody has any source, any research articles, or anything that you think it will be helpful for us, because how many of you listen to our third podcast, like yesterday podcast? Can you please write in chat or unmute yourself and let me know how many of you listen to the podcast? Because yesterday we learned something very interesting. I think Dr. Mohit was here. I think Anis listened to that. Okay, thanks, Anis. So those who are preparing for SMB, it's very important because a lot of like we were scared from the second podcast that maybe it will be difficult for us to take the SMB exam without NGS experience. But yesterday, our guest said it's okay to not have the NGS experience, real life experience, but still you need to learn not only from Lela book. You have to learn from any source. Those who has 5 to 6 or 7 page of NGS, meaning like a deep knowledge of NGS, you have to have. So today let's uh, how can I share the thing? Just give me 1 second. I'm gonna uh, quickly pull into the. I'm gonna share my I think iPad. Maybe that will be helpful to share. Uh, in the meantime, if anybody wants to share anything, share with us. Um, oh, okay, okay. I think we can get one of the PPT and then we can discuss. We can get this from the online. I think everyone should see my screen. If not, just let me know. Um, so this is the thing, and regarding chat, uh, how can I see the chat? Okay, I don't know. I think if anything is going on in chat, just okay. I just we're gonna it will be a very basic discussions, but still, if anybody wanted to share anything or wanted to inform us anything, just share with us. So we know DNA sequencing. Normally, it figuring out the ATGC, and the Sangars is the most important one. I think this is okay to learn, but this kind of history, I think is not for exam. It's for interesting purpose. So this is a basic idea of the history, like how it like the sequencing happened. Um, I think 1990, this one is the most important one. US NIH, it begins large-scale sequencing trials with the Ebola and C. elegans and Saccharomyces cerevisiae. Then first generation sequencing, um, um, like first generation sequencing is the Sanger sequencing and Gilbert, but Sanger sequencing is still a lot of people normally use. Like in New York Genome Center, they have next generation sequencing, but still they use Sanger sequencing to find out any kind of mutation. So still it is very much important. And the basic of this Sanger sequencing is, I think, very important to understand. It's very interesting. I think all of you guys can just watch a video. You will get the idea. But normally, it it is the basic idea of how like it is the base of the sequencing. Um, then second generation sequencing that can do like millions of amplified molecules. This was the drawback of first generation sequencing, is that it is very slow, and it can do very lower amount. Okay. Um, and using a reversible stepwise sequencing chemistry immobilized on a surface. And then comes the next generation sequencing, which is very important. It's we all know high throughput DNA sequencing technique, and plus micro and nanotechnologies, meaning reduced sample size, low reagent cost, and less time. Why next generation sequencing cost low? Because a lot of samples can be done together using the library preparation. So library preparation is another important concept to understand. Massive parallel sequencing sequence thousands of sequences at once and produce enormous amount of data. And the workflow, it is a similar kind of workflow. If you go to the sequencing in the Lala book, it's kind of similar. Um, if anybody knows any other books or anything, please inform us in the chat or share with us what kind of books do you think has the best description of next generation sequencing. Then we can all study that, and we can share knowledge with each other. Um, then normally you need to extract the sample, then DNA fragmentation and in vitro adapter ligation, then clonal amplification by emulsion PCR, and then clonal amplification by base PCR. So if you remember from the chapter 6 of Lela book, there has like this bridge like amplification, which was important for MB, I think. And then pyrosequencing, sequencing by ligation, and sequencing by synthesis. So at first, normally in the NGS workflow, I think those who are preparing for MD, I think this may be very much helpful. I think it will be like kind of helpful for SMB also, creating the DNA fragment, and then adding the platform specific adapter sequence to every fragment. So this is the if this is the sequence, then some of the like uh, another adapter is attached, because by using that adapter it finds it normally binds with the flow cell or beads. Then sequence primer binding sites is added and added barcodes for multiplexing, meaning like another different kind of, like, for suppose 5 or 6 samples, if has it gives it added different kind of like sequence, so that it will be specific for one kind of samples. Then after adapter bindings, then the bridge amplification. This is the flow cells. This is kind of like the whole things which attached with this thing, and then the bridge amplification happened. And then the cluster amplification is done in pre-PCR. So what happens, like, this procedure, it's because I did not do any, like, any of you, if you have like real life experience with NGS, please unmute yourself and share whenever you want. If not, because I'm preparing for to get the real life experience, but I did not have any experience, but I know the basics, and I know the concepts. So I think some of our members said they works with like Illumina and Novasec, so maybe we can get some of the idea from them, like some of the real life experience from them. DNA fragments are flanked with adapter. A solid surface is coated with primer complementary to the two adapter sequence, meaning like this is all the attached with the flow cells. Isothermal amplification with one end of each bridge tethered to the surface, and then clusters of DNA molecules are generated on the chip. Each cluster is originated from a single DNA fragments and is thus a clonal populations. Meaning, like, what I understand is it attached with all the sequence adapt that is connected with this flow cell. It keep attached with the flow cell, and then it increase, like a breeze. It try to like increase like numbers from like these two, it becomes this much of like it increased itself. um, I do not have much knowledge about emulsion PCR, so I'm not sure how what I can say. That if anybody has any knowledge, you can share. I have no knowledge about CRT also. I'm not sure how it works. So sequencing by ligation, that we studied in our Lela book for the MB preparations. So sequencing by ligation is uses the enzyme DNA ligase to identify the nucleotide present at a given positions in a DNA sequence. Uh, for this concept, main important concept is to use the DNA ligase as an enzyme. And how it works is like, suppose the primer is binding, then one of the fluorescence uh, this portion, if it clipped out, then some kind of color, it's gonna give some color, and like in this way, like in every cycle it gives some of the color, and from that color it like it produce some of the colors, and based on that color we can say, okay, this is this for this sequence, this is the uh, A, T, or G, C, like according to the base of like, according to the color we can say, okay, this is the base. Pyrosequencing, another very important topics, I think, for all kind of exams. I think pyrosequencing non-electrophoretic bioluminescence method that measures the release of inorganic pyrophosphate by proportionally converted into visible light using a series of enzymatic reactions. So if you read most of the sequencing, the basic idea is kind of similar. It can produce some light or something, but uh, another interesting thing is like it just changes something, modified something, and try to um, get it done in a new way. So for different kind of like different kind of situation, this kind of things can be used, like different things. So for this pyrosequencing, it used the inorganic pyrophosphate. And when inorganic pyrophosphate produced, that inorganic pyrophosphate uh, uh, produce light from the luciferase, like this ATP produce light from luciferin using the luciferase enzyme to oxyluciferin. Okay, so that's how the light is producing, and nucleotide incorporation generates light seen as a peak in the pyrogram graph. And I think this kind of pyrogram, like to understand this, is very much important. Uh, like if you see one graph, you should understand, okay, what should be the sequence. Single molecule real time sequencing. Uh, I think there has some right now. It's they modified this real time like kind of like PCR to digital PCR kind of things, and it's kind of like similar for sequencing maybe somehow. So single molecule real time is a parallelized single molecule DNA sequencing method. A single DNA polymerase enzyme is affixed at the bottom of a JMW with a single molecule of DNA as a template. So I think what happens is like whenever this sequencing, like normally what we understand from real time is that it's gonna give us the real time scenario of the run. So I think according to my concept, maybe this is the idea of single molecule real time sequencing, which is in every step it will show you what is the sequence. So that is
Abu,
yes, yes.
this is not real time. SMRT is for it. It will be used for pack bio.
Mm-hmm.
Okay, and it is for single read sequencing. It's like long read sequencing. Okay, so this is see main thing you should know what are short reads, what are long reads, and which chemistry. I think the question will maybe I ask which chemistry which used for which machine. For example, Illumina is synthesis by sequencing. Okay, synthesis. Thermo is by pH method, and this SMRT for pack bio. So this one is not it's a real time, but it's a single reads, long reads. Okay.
So is it a kind of similar as isosequencing, like that 2 by PacBio,
No,
meaning like long
yeah,
read?
it's a long reads for pack bio. Okay.
Okay, okay, okay. Now it
And
makes sense. Okay, okay. Got that.
only for mainly for de novo sequencing. You know what is de novo sequencing?
De novo sequencing is, I think, like de novo is the production something
No, no,
new.
no, no, no. De novo is like when you don't have the reference sequence, like so de novo assembly. Okay, for example, now some plants or some animals, there's no reference sequences. Okay, have reference sequences, so you do long read sequencing that we use pack bio. Okay.
Okay, got that. So meaning
Okay.
like,
Yeah, and yeah, so that they use for like some plants which is very endogenous. For example, someone want to research on like new sequencing, new generation of sequence which they will submit to the NCBI on all and all. Okay, that time they use this pack bio.
okay, okay, got that. That makes
Yeah,
sense. Yeah.
Illumina is very short read sequencing. It's a short read. It's less than 100, around 150 base pair short reads.
Yes,
Okay.
yes. 70 to 150 they say normally, right?
Yeah, short read sequence, and it's a bi-direction sequencing. It has two reads, R1 and R2. Okay. Uh, whether about Thermo Fisher ion torrent, it's a single read they use R1 only. Okay, and
Got
it's by
that.
pH based synthesis. So
Okay.
it's change in pH.
Hmm.
And, and this pyrosequencing, I think Roche used that FIFO sequencing or solid ligation by solid and ligation sequencing used by solid machine.
Okay. So ligation with solid machine,
Yeah.
pyrosequencing by Roche,
Yes.
SMRT by PacBio.
As a bio, the bridge amplification, that is which you showed, that is for Illumina. Okay.
Hmm.
And and ion torrent, that is by pH, is for ion torrent. Okay,
Okay. pH
and
is by ion torrent. Got that.
ion torrent, change in the pH when there is different, each nucleotide will be added. There will be read, the pH will be read, and pyrosequencing mainly used for methylation.
Hmm.
Methylation, this is called methylation PCR, right? Or methylation sequencing. That is called bisulfate sequencing. What about
Hmm.
bisulfate sequencing?
Yes.
Bis- Bisulfate conversion of DNA. So before sequencing, there will be conversion of DNA by bisulfate enzyme
Hmm.
that we will use what do you call, to check the any methylation CPG island.
Hmm. Got that.
Okay, you
That
can
is
continue.
okay.
Anything is
Okay.
there?
Okay. Thank you so much. I think it was very much informative because I just saw this whatever you just saw. I was gathering that kind of knowledge, but now it makes more sense. Like all the differences from these different companies, what they are using.
Yes.
It will be very much helpful. I think those who are participated, it will be very much helpful. Those, especially those who gonna take the MB, MB questions, they ask this kind of questions, and this kind of clarification gonna be helpful for them. Whatever you just said, it is everything in the Lela book. I think those who gonna read that now they will understand more. Uh, I think here is all the commercially available technologies: Illumina, Solexa, Roche, 454, Helicoach Bioscience, Life APG Solid, Pacific Bioscience, and Ion Torrent Technologies. Um, and NGS normally differs from each companies as we just discussed. So Illumina use the solid phase amplification, which can produce 100 to 20 million specially separated clusters, providing free ins to which universal sequencing primer can be hybridized to initiate the NGS. So it is the most commonest. Um, Normally, people do that by 5 days. They say 1 to 10 days, but 5 days they can do the whole genome sequence here. Uh, 0.05 to $0.04 per MB. That's very cost effective. Application DNA sequencing, gene regulation analysis, and all kind of like cytogenetic chip sequencing, small RNA discover analysis. Uh, I think we don't need that much information. Roche used the sequence much longer reads by sequencing multiple reads at once by reading optical singles as bases are added, and the DNA or RNA is fragmented into shorter reads up to 1 kb, meaning it's bigger than that Illumina short read sequence. Uses emulsion PCR. So Roche is using normally emulsion PCR and pyrosequencing as a sequencing approach. I think cluster amplification, you will find that in chapter 6 of Larapo. Uh, then all of the sequencing reads we get from 454 will be different lengths, meaning from that machines, because different numbers of these bases will be added with each cycle. And application can be whole genome sequencing, targeted sequencing, sequencing based transcriptome analysis, and metagenomics. Um, I think regarding ion torrent, and I think we will have some more, because I think I have another research article or some other uh, place. I got that, but I have seen that, but I did not get that. I could not prepare for today's session, but maybe next week we can discuss more about ion torrent, and we can get more in-depth knowledge, and try to get the Sanger and AGS. So this is the all the comparison also of all the NGS platforms, but we wanted to go in depth of NGS. This was a basic kind of idea, but those who are for SMB, I think it requires a little bit more of in-depth uh, knowledge. So next week we will try to get a little bit more information. Anybody wanted to share anything? If not, then thank you so much for joining. It was a brief discussion. I was a little bit busy, so I could not prepare much, but maybe next week we will be able to discuss a little bit more Okay, so Dr. Muid, I think I just said that like this week I was little bit busy, as you guys know, with the podcast and everything, so I could not prepare much. But next week I think I can prepare more, and um, um, I think you will have some exam or something. But in the meantime, if I find anything that is interesting, I will share all of with you guys. Please check the Discord for any kind of research article or anything that I got. I will share with you. Okay.
Sure, sure. Maybe in after my exam,
Mm-hmm.
I'll give some presentation. Inshallah. Because,
Yeah, inshallah.
this will be busy in preparing because next 23rd I have exam. Okay.
Okay, okay. We all pray for you, and hopefully everything will be fine. And we hope everything will be go well. And yes, please, we'll be happy to listen to your experiences. Anybody
Sure,
wanna add
sure.
anything? If not, then thank you so much for
Yeah,
joining.
just I want to add. Just I want to add.
Mhm.
Like,
Yes, yes, sure.
see for any NGS, you should know for what technology you any. It's all based on your applications. Which technology you want to use for for example, disease identifying mutation. Which platform you want to use for to do research. Which platform you use for to identify the somatic mutations. So
Mhm.
all this properly, I think you should know. For example, Illumina has the highest rate of accuracy,
Mhm.
uh, but com- but compared to NGS, Sanger sequencing has highest rate of accuracy, right? Because when you use NGS like Illumina to confirm the mutations in the father or in the mother or in the proband, you should use Sanger sequencing. Uh, Without Sanger sequencing, you can't confirm the NGS result. Okay. So when you have the NGS report, first thing they will ask to do the confirmation by Sanger sequencing, because
Mhm.
sometimes what happen in NGS, the reads will be low.
Mhm.
So for me, maybe 30x or maybe 45x. It
Mhm.
depends on the coverage, how much depth the NGS has been done. So everybody knows about this coverage and depth, right?
Yes, I think there has the percentage. The coverage means coverage means like, okay, if it is giving like 50% for A or something like 58% for A and T for, so that according to that percentage, like A will be on
Yeah.
that position, something like that, right?
Yeah, it's no yeah each reads will have the each when why we call the it has multiple reads, right? It has like millions of reads when you get the one sample sequencing. Okay, so maybe one sample will give maybe 70 million reads. Okay, so each genes has different types of no million of reads. So for example, each gene maybe it's just 30x or 40x or sometimes 120x. Okay, 120x means 120 times the reads has been run. Okay, okay. For example, if you find the mutation in BRCA1 in specific exon and specific location, okay. In that, for example, if you say 80x means 80 times it has run. Okay, has been in that location. In that, if you change in A to T, for example, as you said, if it is heterozygous mutation, maybe 80%, sorry, 45% of reference sequence or 55% of mutant sequence. Okay, in case of heterozygous, if it is homozygous mutation, it will be 100% the mutant allele. Okay,
Got that, got that.
you got it. And
Yes.
but in case of somatic mutations, the allele variant sequence will be very low.
Hmm. So for like homozygous mutation, it will be 100%. For heterozygous, it will be like 50 to 60% you said, and for a somatic mutation, it will be very low percentage.
If it is somatic mutations, like we give the threshold of zero point 0.5 to 1 allele frequency. Okay. Then in maybe after one, I'll explain maybe now after
Mhm.
my exam I will explain how to interpret the whole exome data, and because I have more experience on whole exome sequence analysis and interpretations, how to look the IGV and all.
Hmm. That will be very much helpful. Yeah.
Okay,
And
okay.
Yeah, sure. Thank you so much. And in the meantime, if you think you have some of the research articles or some of the books or chapters that will be helpful for us to read in this week, please share with us in the Discord. Just upload that or send me. Um,
I'm
uh,
outside now. Okay.
yeah, sure. No problem.
Sure, I will.
Not now. Whenever you are available. Yeah, please.
Yeah. So when
Just
I'm available, we will have a meeting maybe
yeah,
next
yeah.
week, inshallah. Okay.
Yeah, okay, inshallah. Thank you so much. Thank you everyone for joining,
Thanks.
and thank you so much for nice input, Dr. Min.
Yeah.
And
Thank you.
we pray for you. Thank you. Bye bye.